The overall goal of this protocol is to describe an in vitro surrogate biosensor for testing microglial response to nanoparticles. We will then determine neurotoxicity following exposure to conditioned media from microglia stimulated with nanoparticles.
This method can help answer key questions in the neuroscience field, such as how to rapidly screen nanoparticles for neurotoxicity.
The main advantage of this technique is that our model provides the ability to directly activate microglia and determine if secreted factors are neurotoxic without the use of expensive, long-term rodent studies. Though this method can provide insight into microglial-induced cell death following nanoparticle exposure, it can also be applied to other models of neurodegenerative diseases within the context of testing potential neurotoxic environmental pollutants.
Generally, individuals new to this method will struggle with the conditioned media filtration step, because the filtration efficiency may differ depending on the size, shape, and the concentration of the nanoparticles.
In preparation, warm the DMEM supplemented with 10% FBS and 1% PSN to 37 degrees Celsius. Then take frozen BV2 microglial cells obtained from passages 18 to 25 and rapidly thaw them at 37 degrees Celsius. Next, gently transfer the thawed cells to a 75 square centimeter vented flask containing 10 milliliters of medium and incubate the flask. After 24 hours, replace the medium and continue growing the cells until they reach a confluency of